Have you ever really stopped to think about what's happening inside your cells? Like right this second?
It's kind of mind-boggling when you actually consider the scale and the complexity, isn't it?
Totally. I mean, we're talking millions, literally millions of molecules constantly being made, sorted, delivered.
All happening within this incredibly tiny space. It's like a city that never sleeps, but microscopic.
Yeah, smaller than a speck of dust. And somehow it all just works. It's like this perfectly orchestrated ballet, zero traffic jams.
It really is an unparalleled feat of organization because, under a basic light microscope, it just looks like, well, mostly empty space, maybe a few blobs.
Right, kind of like a blurry, structureless soup.
Exactly. But that view is so misleading. It's anything but simple.
And that's why today we're doing a deep dive into cytoplasmic membrane systems, structure, function, and membrane trafficking.
We're going inside the eukaryotic cell, basically.
Yeah, our mission is to really unravel this intricate inner world. We're looking at those dynamic membrane-bound compartments, how they all work together.
And importantly, how we even figured all this stuff out, the discoveries behind it.
Exactly. We're leaning heavily on Karp cell and molecular biology as our guide here. It's a fantastic resource for this.
And here's a really fascinating angle to start with. Think about viruses. The
Right.
cell's interior is this incredibly complex protected environment, right? A fortress.
Sure.
And yet viruses have evolved these incredibly clever ways to hijack the whole system.
Hijack it how?
They use the cell's own membrane systems, the plasma membrane, endosomes, Golgi, not just to get in, but also to build their own little virus factories inside.
Wow, like commandeering the production line.
Precisely. For replication, making more viruses, and then getting back out again. It just highlights how central and, well, how exploitable these internal membrane highways are.
Okay, so let's trace how our understanding evolved, this inner highway, the endomembrane system. It wasn't always clear it existed, was it?
Not at all. Before the 1940s, before the electron microscope really came into its own, the cytoplasm just looked, well, devoid of structure was the term often used.
Just that blurry soup we mentioned.
Exactly. Then the electron microscope arrived, and boom, everything changed.
Suddenly you could see.
What? Suddenly, researchers could resolve this incredible, diverse array of membrane-bound structures. Tiny vesicles, these complex interconnected channels we now know as the ER.
And those stacks of sacs. The Golgi.
Yep, the Golgi complex. It was a visual evolution. It showed this hidden world of complexity of actual structures inside the cell.
And crucially, scientists realized these weren't just random bits and pieces floating around.
Right. They realized it's a functionally integrated network. That's the key, integrated.
The endomembrane system. So that includes, let's
Okay, you've got the endoplasmic reticulum, or ER, the Golgi complex, endosomes, lysosomes.
and all the little transport vesicles moving between them.
Exactly. They're all connected, functionally speaking, constantly exchanging materials.
You can think of it like a cellular superhighway system, right?
Yeah.
With traffic going both ways.
That's a great analogy. First, you have the biosynthetic pathway, sometimes called the secretory pathway.
Okay, biosynthetic, so making things and sending them out.
Precisely. Materials flow out from where they're synthesized. We're taking proteins, lipids, complex sugars, moving from the ER, then through the Golgi.
And then off to various destinations.
Yep. Places like lysosomes, endosomes, maybe secretory vesicles to be stored, or vacuoles in plant cells. Or they go straight to the plasma membrane to become part of it, or even get secreted completely outside the cell. That last bit is exocytosis.
And within that outward pathway, there are different styles of delivery, aren't there?
Right. There's constitutive secretion. Think of this as the default, continuous flow, nonstop.
Like routine maintenance.
Sort of, yeah. It's essential for constantly supplying materials to build things like the extracellular matrix outside the cell, or just maintaining the plasma membrane itself. It's always on.
Okay, that makes sense. What's the other one?
The other is regulated secretion. This is much more specialized. Materials, think hormones, digestive enzymes, neurotransmitters, they get packaged up into these membrane-bound containers, often large, densely packed things called secretory granules.
So they're stirred, waiting for a signal.
Exactly. They're only released when the cell gets a specific stimulus, like a nerve impulse triggering neurotransmitter release, or a hormonal signal causing an endocrine cell to secrete its hormone. It's secretion on demand.
Got it. So that's the out pathway. What about traffic coming in?
That's the endocytic pathway. Here, materials flow in from the cell surface, moving towards the interior.
Where do they usually end up?
Typically, they journey to endosomes and then often onto lysosomes, where they can be broken down, degraded, recycled. It's the cell's way of taking things up from the environment or internalizing signals.
So a balanced flow. Stuff going out, stuff coming in. But how does the cell keep it all straight? How does protein X know to go to the lysosome while protein Y needs to be secreted?
Ah, that's where the sorting signals come in. These are like molecular ZIP codes, or maybe address labels.
And coded right into the molecule.
Usually, yes, either in the amino acid sequence of a protein or sometimes in the carbohydrate chains attached to it. Specific sequences act as signals.
And these signals are recognized somehow.
Yep. By specific receptors, often located in the membranes of the budding vesicles. These receptors grab the cargo with the right signal, ensuring it gets loaded onto the correct transport vesicle heading for the right destination. It's incredibly specific.
Okay, that's the overview. But how on earth did scientists figure all this out? Going from just seeing fuzzy blobs to understanding these pathways and signals.
Right. It wasn't easy. Seeing the structures was one thing, understanding the function, the dynamics. That required some really clever experimental breakthroughs. This is where the story gets exciting.
Let's hear about them. What were the key techniques?
One of the absolute foundations was autoradiography. This was developed quite early on.
Autoradiography. Sounds like taking pictures of radioactivity.
That's basically it. You introduce radioactively labeled molecules, say, amino acids, into cells. Then you use photographic emulsion placed over the cells or tissues. Wherever the radioactive material is, it exposes the emulsion, like light-hitting film.
So you could see where the labeled molecules went.
Exactly. You get a snapshot of their location.
And this led to the pulse chase experiment. That sounds famous.
It is. Groundbreaking work by James Jamieson and George Pollard, studying pancreatic cells, which secrete lots of digestive enzymes.
What did they do?
They gave the cells a brief pulse, a short exposure to radioactive amino acids. These get incorporated into newly synthesized proteins almost immediately.
Okay, so you've labeled the proteins just made.
Then comes the chase. They washed away the radioactive medium and replaced it with normal, unlabeled medium. Now, those labeled proteins start their journey through the cell.
And they could track them over time using autoradiography.
Precisely. They took samples at different time points after the chase began. They saw the radioactive signal first appear in the rough ER,
where proteins are synthesized.
Then, a bit later, the signal moved to the Golgi complex.
Processing and packaging.
Then into secretory granules near the cell surface. And finally, the radioactivity appeared outside the cell as the proteins were secreted.
Wow. So they literally mapped out the entire secretory pathway step by step.
They defined it. It was the first direct evidence of this sequential flow. ER to Golgi to secretory vesicles to outside. Just brilliant.
Okay, that's amazing for tracking movement in fixed cells. But what about seeing it live?
Ah, that's where green fluorescent protein, or GFP, completely revolutionized things. Game changer.
Right, the stuff from jellyfish that glows green. How did that help?
Researchers could genetically fuse the GFP gene to the gene of a protein they wanted to study. Now, the cell makes this fusion protein, and it glows.
You can watch it move inside a living cell in real time.
Exactly. No fixing, no radioactive labels needed. You just put the cell under a fluorescence microscope and watch.
That must have opened up so many possibilities.
Immense. For example, people infected cells with a modified virus, vesicular stomatitis virus, VSV, that made a viral coat protein fused to GFP.
So the viral proteins glowed.
Yep. And they could watch these fluorescent proteins be made in the ER and then trafficking synchronously, like a wave, through the Golgi and out to the plasma membrane.
Wow. And I read something about temperature sensitivity being used here, too.
Yes, that was another clever trick. They used a mutant version of the viral protein, VSVG, that couldn't fold properly and leave the ER at a higher temperature, say 40 degrees Celsius.
So it gets stuck in the ER.
Right, it accumulates there. Then you just lower the temperature to, say, 32 degrees. Now the protein can fold correctly and exit the ER.
And because it all accumulated first.
It moves out as a synchronized wave. It makes it much easier to observe the progression through the different compartments, ER to Golgi to plasma membrane. Really elegant way to visualize the pathway live.
Okay, autoradiography, GFP. What else? How about taking the cell apart?
Ah, yes. Subcellular fractionation. This was pioneered back in the 50s and 60s by researchers like Albert Claude and Christian Duduve, another Nobel-winning contribution.
How does that work? You basically blend the cells.
Essentially, yes. You carefully break the cells, open homogenization. This fragments the different membrane systems, like the ER and Golgi, into tiny, sealed, spherical vesicles called microsomes.
Little bubbles of membrane.
Exactly. And the key is that microsomes derived from different organelles, like rough ER versus smooth ER versus Golgi, have different sizes, densities, and properties.
So you can separate them.
Yep, using centrifugation. You spin the mixture at different speeds, and the different types of microsomes pellet out at different points. You can isolate fractions enriched for rough ER, smooth ER, Golgi, etc.
And then study what's inside them.
Precisely. Early on, they could analyze the enzyme activities. Now, with modern proteomics and mass spectrometry, we can identify hundreds, even thousands of different proteins in an isolated organelle fraction.
So you get a complete molecular inventory.
You get a comprehensive molecular portrait, as Karp puts it, like identifying over 160 distinct proteins just in the membrane of a phagosome, for instance. It tells you what machinery is present in each compartment.
And can you get these isolated bits to actually do something?
Sometimes, yes. That's the idea behind cell-free systems. You take the isolated components and see if they can perform their functions in vitro in a test tube.
Like what?
Palad and Sikovits, for example, showed that isolated rough microsomes, bits of rough ER, could actually take up newly synthesized secretory proteins into their internal space, their lumen.
Well, proteins made on free ribosomes couldn't get in.
Right. Those just stayed in the surrounding fluid. It proved the ER membrane itself was involved in sequestering these proteins during synthesis.
And this led to work on vesicle transport, too.
Huge advances there. James Rothman and Randy Schekman, more Nobel laureates, used cell-free systems extensively to dissect vesicle budding and fusion. They could even reconstitute vesicle budding from artificial lipid membranes, liposomes, just by adding purified coat proteins. Incredible detail.
Amazing. Okay, another powerful approach must be studying things when they go wrong, right? Mutants.
Absolutely. Looking at mutant phenotypes. If a gene is mutated, the protein it codes for might be defective, leading to a specific block in a cellular process. Randy Schekman's work again, this time with yeast genetics, was pivotal.
Yeast? Why yeast?
Yeast are simple eukaryotes, easy to grow and manipulate genetically. Schekman isolated a whole collection of yeast mutants called sec mutants because they were defective in the secretory pathway.
What did they show?
Each mutant had a block at a different step. For example, a Sec12 mutant couldn't form transport vesicles from the ER, so its ER became hugely expanded and accumulated proteins.
A traffic jam at the ER exit.
Exactly. Another one, Sec17, accumulated lots of unfused vesicles because it had a defect in the fusion step at the Golgi. By analyzing these different blockages, they could piece together the sequence of events and identify the genes and proteins required for each step.
And does this yeast work apply to us?
Remarkably well. It turned out that the fundamental machinery of membrane trafficking the proteins involved in budding, fusion, targeting is highly conserved across evolution, from yeast to humans. You can often swap the human protein for the yeast one, and it works.
Wow. Okay, one more technique from the book. RNA interference? RNAi.
Right, RNA interference or RNAi. This is a more recent technique, but very powerful for functional studies.
How does it work? It silences genes, right?
Essentially, yes. Cells have a natural mechanism where small RNA molecules, called siRNA, small interfering RNAs, can bind to specific messenger RNAs, mRNAs, and trigger their degradation or block their translation into protein.
So you can specifically shut down the production of almost any protein you want.
Exactly. Researchers create libraries of siRNAs targeting thousands of different genes. They can then introduce these into cells, one by one or in pools, and see what effect silencing each gene has on a particular process they're studying.
Like transport through the Golgi.
For instance, yes, they might look for siRNAs that cause a Golgi enzyme, like mannose 2, to get stuck in the ER instead of reaching the Golgi. If silencing gene X causes that phenotype, then protein X is likely involved in ER to Golgi transport.
And it's easier than making mutants.
Often, yes. Especially in mammalian cells, where generating targeted gene knockouts can be more complex and time-consuming. RNAi provides a relatively rapid way to screen for genes involved in a specific pathway.
Okay, armed with all those techniques, let's dive into the structures themselves, starting with the endoplasmic reticulum, the ER. You called it the manufacturing hub.
It really is. It's this huge, sprawling network of membranes, interconnected tubules, and flattened sacs called cisternae that extends throughout much of the cytoplasm.
And it encloses its own internal space.
Yes, the ER lumen, or cisternal space. It's continuous throughout the network, but distinct from the surrounding cytosol.
And it has two main parts we mentioned, rough and smooth ER.
Right. The rough ER, RER, Gets its name from the ribosomes studded all over its cytosolic surface. It typically looks like interconnected, flattened sacs, and it's often continuous with the outer membrane of the nuclear envelope.
Ribosomes, meaning protein synthesis happens there.
Exactly. And the smooth SER, lacks ribosomes, so its surface looks smooth. It tends to be more tubular and network-like.
Are they completely separate?
No, their membranes are continuous. Proteins and lipids can, in principle, diffuse between the RER and SER. However, they definitely have distinct protein compositions tailored to their different functions.
Okay, let's talk functions. What does the smooth ER do?
The SER has several key roles, and it's particularly abundant in certain cell types.
Like what?
Like cells that synthesize steroid hormones, think adrenal cortex, testes, ovaries. The enzymes for steroid synthesis are located in the SER.
Okay, hormone production. What else?
Detoxification, especially in the liver. The SER contains enzymes, like the cytochrome P450 family, that metabolize various organic compounds, drugs like barbiturates, ethanol from alcoholic drinks. They make them more water-soluble for excretion.
Is that why chronic alcohol or drug use can affect the liver?
Yes, and interestingly, chronic exposure can lead to proliferation of the SER in liver cells, increasing the capacity for detoxification. This is why tolerance develops.
Fascinating.
Yep.
Any other major SER roles?
Calcium storage. The SER membrane has pumps that actively sequester calcium ions from the cytosol into the ER lumen. This is incredibly important in muscle cells, where the specialized SER is called the sarcoplasmic reticulum.
For muscle contraction, right? Release of calcium triggers it.
Exactly. The regulated release and reuptake of calcium by the sarcoplasmic reticulum controls muscle contraction and relaxation.
Okay, that's the smooth ER. What about the main jobs of the ref ER?
The RER is really the entry point, the starting point, for the entire biosynthetic secretory pathway.
So making stuff that's going somewhere else.
Precisely. It's the site of synthesis for basically all proteins that are destined to be secreted from the cell, inserted into cellular membranes, plasma membrane, Golgi, lysosome membranes, or reside within the lumen of the Golgi, endosomes, or lysosomes.
That's a lot of proteins.
It is. About 1/3 of all proteins in a typical mammalian cell start their life on the RER. It's also involved in the initial steps of adding carbohydrate chains, glycosylation, and in the synthesis of phospholipids for membrane formation.
You mentioned secretory cells often look polarized.
Yes, cells specialized for secretion, like pancreatic acinar cells making digestive enzymes, or goblet cells making mucus, often have a very distinct internal organization.
How so?
The RER is typically concentrated near the base of the cell, basal surface. The Golgi complex is usually located more centrally or apically, and the secretory granules containing the final product accumulate at the apical surface ready for release. It physically reflects the flow of material. RER Golgi granules fat
A visible production line. So let's clarify protein synthesis location. We said some are made on RER ribosomes, some on free ribosomes in the cytosol. Can we break that down again?
Sure, it's a fundamental distinction. Ribosomes attached to the rough ER synthesized.
Oh.
Secreted proteins, integral membrane proteins, the ones embedded in membranes, and soluble proteins that will end up inside the organelles of the endomembrane system, ER lumen, Golgi lumen, lysosome lumen, etc.
And this happens as they're being made.
Mostly, yes. It's primarily co-translational translocation. The protein starts entering the ER while it's still being synthesized by the ribosome.
Okay. What about the free ribosomes floating in the cytosol?
They synthesize proteins destined for different locations. Proteins that will stay in the cytosol itself, like enzymes for glycolysis, cytoskeletal proteins, peripheral membrane proteins, which associate with the surface but aren't embedded, and proteins destined for the nucleus, mitochondria, chloroplasts in plants, and peroxisomes.
And these get imported after they're made?
Generally, yes. That's post-translational import. The fully synthesized protein is released into the cytosol and then targeted and imported into the correct organelle. Different mechanism.
Let's focus on that co-translational process into the ER. How does a ribosome know to go to the ER in the first place?
It starts with a protein being synthesized. If it's destined for the ER pathway, the first part of the protein sequence to emerge from the ribosome is a specific signal sequence.
A little tag at the beginning.
Usually, yes. Typically 1530 amino acids, often with a stretch of hydrophobic residues. This signal sequence is recognized by a complex called the signal recognition particle, or SRP.
Okay, SRP sees the signal, then what?
The SRP binds to both the signal sequence and the ribosome, and this interaction actually pauses translation temporarily.
Halts protein synthesis. Why?
To give the whole complex SRP, ribosome, partially made protein time to find the ER membrane, the SRP then targets the complex to an SRP receptor located in the ER membrane.
Docking at the ER.
Right. Once docked, the ribosome is handed off from the SRP complex to a protein channel in the ER membrane called the translocon.
The gateway into the ER.
Exactly. The signal sequence inserts into the translocon channel, the translation pause is released, and now, as the rest of the polypeptide chain is synthesized, it's threaded directly through the translocon channel into the ER lumen.
And the signal sequence, does it stay on?
Usually not for soluble proteins. There's an enzyme called signal peptidase in the ER lumen that often cleaves off the signal sequence once the protein is inside.
Wow. And you mentioned GTP binding proteins are involved.
Yes, G proteins play crucial regulatory roles here. Both the SRP and the SRP receptor are GTP binding proteins. They act like molecular switches, cycling between an active GTP bound state and an inactive GTP bound state. GTP binding and hydrolysis control the key steps like SRP binding, targeting, and release, ensuring the process is directional and properly timed.
Okay, that explains how soluble proteins get into the ER lumen. What about proteins that are meant to stay in the ER membrane? Integral membrane proteins.
They use the same initial machinery signal sequence, SRP, SRP receptor, translocon.
So they start entering the translocon too.
Yes, but the key difference lies in their hydrophobic transmembrane segments. These are stretches of the polypeptide that are destined to span the lipid bilayer.
So how do they get out of the aqueous channel and into the oily membrane?
The translocon itself is thought to function like a lateral gate. When a transmembrane segment enters the channel, the translocon can actually open sideways, allowing that hydrophobic segment to partition directly out of the channel and integrate into the surrounding lipid bilayer.
So the translocon threads some parts through and shunts others sideways into the membrane.
That's the model. It can handle multiple transmembrane segments, opening and closing its lateral gate to stitch the protein into the membrane in the correct orientation as it's being synthesized.
That's incredibly sophisticated. And this process also builds the membrane itself.
Well, it inserts the proteins. Membrane lipids are also synthesized primarily in the ER, and a key point is membrane asymmetry.
Meaning the two sides of the membrane are different.
Exactly. The two leaflets, the inner cytosolic and outer luminolexoplasmic layers of the lipid bilayer, have different phospholipid compositions. This asymmetry is established in the ER and is maintained as membranes bud off as vesicles and fuse with other compartments.
So the side facing the cytosol always stays facing the cytosol.
Always. And the side facing the ER lumen will eventually become the side facing the outside of the cell if it reaches the plasma membrane, or the inside of a lysosome or Golgi lumen.
And carbohydrates are added in the ER too, right?
Yes.
Glycosylation.
Yes, a major event in the rough ER is N-linked glycosylation. Almost all proteins synthesized on the RER get carbohydrate chains attached to them.
Becoming glycoproteins. Why? What do the sugars do?
Several things. They can act as binding sites for other molecules, they play a critical role in ensuring the protein folds correctly, and they can help protect the protein from degradation.
And how are they added? One sugar at a time.
Actually, no. That's what's really unique about N-linked glycosylation. A large, preformed branched oligosaccharide block containing 14 specific sugar residues, N-acetylglucosamine, mannose, and glucose, is first assembled.
Assembled where?
It's built step by step on a specialized lipid carrier molecule embedded in the ER membrane called dolichal phosphate.
Okay, so this big sugar tree is built on a lipid anchor.
Right. And then this entire pre-assembled block of 14 sugars is transferred en bloc all at once from the dolichal phosphate carrier onto specific asparagine N residues within the polypeptide chain as it's entering the ER lumen. That's why it's called N-linked.
Wow. Transferred as a whole unit.
It's quite a feat of molecular engineering.
And you mentioned this can go wrong in human diseases.
Yes. Defects in this glycosylation pathway cause a group of serious inherited disorders called congenital diseases of glycosylation, or CDGs.
Like the one treatable with mannose.
Exactly. CDG1B. It's caused by a deficiency in an enzyme called phosphomannose isomerase. Patients can't properly synthesize one of the precursor sugars needed for that dolichal-linked oligosaccharide. It used to be fatal.
But now,
simply giving these patients oral mannose supplements can bypass the metabolic block, allowing glycosylation to proceed more normally. It turns a devastating disease into something manageable, a really stunning example of basic science leading to effective therapy.
incredible. Okay, so proteins are entering the ER, getting sugars added. Does the ER check if they're folded correctly?
Oh, absolutely. The ER has a very sophisticated quality control system to ensure proteins don't leave unless they're properly folded and assembled.
How does that work, especially for these glycoproteins?
It involves those N-linked oligosaccharides. After the initial block of 14 sugars is added, some sugars are quickly trimmed off, particularly glucose residues. If a glycoprotein still has one terminal glucose remaining, it can bind to ER chaperones.
Chaperones like heat shock proteins, helpers for folding.
Exactly. Key ER chaperones involved here are calnexin and calreticulin. They bind to these monoglucosylated glycoproteins and give them time to fold properly, preventing aggregation.
What if it still doesn't fold right?
Then, an amazing enzyme called UGGT acts as a confirmation sensor. If UGGT recognizes that the protein is still misfolded, it specifically looks for exposed hydrophobic patches, it will actually add a glucose back onto the oligosaccharide chain.
Really? So it can bind the chaperones again?
Precisely. It gives the protein another chance to bind calnexin or calreticulin and try folding again. This cycle can repeat multiple times.
A built-in proofreading and refolding cycle. But what if it just keeps failing?
If, after several attempts, the protein remains stubbornly misfolded, eventually a different enzyme will trim off a specific mannose residue from the oligosaccharide chain. This mannose trimming acts as a signal.
A signal for what?
A signal that this protein is terminally misfolded and needs to be destroyed.
Destroyed where?
Yeah.
Still in the ER.
No, this leads to a process called ER-associated degradation, or ER. The terminally misfolded protein is targeted for retrotranslocation, meaning it's actually transported backwards out of the ER through a channel, possibly the translocon again or a related one, into the cytosol.
Kicked out of the ER.
Yep. Once in the cytosol, its oligosaccharide chains are usually removed by an enzyme called N-glycanase. It gets tagged with ubiquitin molecules, and then it's degraded by the proteasome, the cell's major protein destruction machinery.
Wow, so the ER catches errors, tries to fix them, and eliminates the failures.
It's a very rigorous quality control checkpoint.
And you mentioned cystic fibrosis connection.
Yes, EROD is implicated in quite a few human diseases. In many common forms of cystic fibrosis, the mutation is in the CFTR protein. The mutation causes misfolding, but the protein, if it could reach the plasma membrane, might still have some residual function.
But ER catches it.
But the ER quality control system recognizes it as misfolded and targets it for ERAD. So it gets destroyed in the cytosol and never reaches the cell surface where it's needed to function as a chloride channel. The disease isn't caused by a complete lack of the protein, but by its failure to pass ER quality control.
Yeah, it's a crucial distinction. Okay, what if the ER gets completely overwhelmed with misfolded proteins? If ERAD can't keep up?
That triggers a cellular stress response called the unfolded protein response, or UPR.
Trying to cope with the backlog.
Exactly. The cell senses this accumulation of unfolded proteins in the ER lumen. Special sensor proteins embedded in the ER membrane get activated. Normally, they're kept inactive by binding to chaperone proteins like BiP.
But if BiP is busy dealing with unfolded proteins,
then BiP releases the sensors, allowing them to become active.
what do the activated sensors do?
They initiate signaling pathways that have several major consequences. One is to increase the synthesis of more ER chaperones, folding enzymes, and components of the ER machinery, basically, boosting the ER's capacity to handle the problem.
Trying to scale up the cleanup crew.
Right. Another major effect is to temporarily decrease overall protein synthesis in the cell, reducing the load of new proteins entering the ER. This is often done by phosphorylating a key translation initiation factor called EIF2-alpha.
So slow down production while increasing cleanup capacity.
That's the idea. The UPR is fundamentally a pro-survival response, trying to restore homeostasis in the ER.
But can it fail?
Yes. If the stress is too severe or prolonged and the cell can't resolve the problem, the UPR pathway can actually switch gears and trigger programmed cell death, apoptosis. It's like the cell decides it's better to self-destruct than risk malfunctioning.
A double-edged sword, then.
Very much so.
Okay, assuming a protein folds correctly, passes quality control, how does it get from the ER to the next stop, the Golgi?
It exits the ER from specialized regions called ER exit sites, or ERs. These are distinct patches on the ER membrane, usually on the transitional elements of the RER, and they notably lack ribosomes.
So specific launch pads.
Right. At these sites, transport vesicles butt off, capturing cargo destined for the Golgi and beyond. These first vesicles are typically coated with a protein complex called CoPII.
We'll come back to coats later. What happens to these initial vesicles?
They don't seem to travel individually all the way to the Golgi. Instead, they quickly fuse with each other near the ER, forming larger structures called vesicular tubular carriers, or VTCs.
Okay, bigger transport units.
Exactly. These VTCs make up what's called the ER-AJC, the ER-Golgi intermediate compartment. It's a collection of membranes situated between the ER and the Golgi. These VTCs then actively move towards the Golgi complex, typically along microtubule tracks like trains on a railway.
Delivering their cargo to the Golgi entrance.
Right, delivering it to the cis face of the Golgi stack.
Okay, let's talk about the Golgi complex, the cell's processing plant and sorting center.
That's a perfect description. It's typically composed of a stack of flattened membrane-enclosed sacs called cisternae. Think of a stack of pancakes, but hollow.
And it's polarized, right? Different ends.
Yes, very much so. It has a distinct polarity. The cis entry face is oriented towards the ER. This side includes a network of tubules and vesicles called the cis-Golgi network, CGN, which receives the material arriving from the ER-GC.
That's the receiving duct.
Then you have the medial cisternae, which form the main body of the stack. And finally, the trans-exit face points away from the ER towards the plasma membrane. This side includes the trans-Golgi network, TGN.
The shipping department.
Exactly. The TGN is a major sorting station where proteins and lipids are sorted and packaged into different types of vesicles heading to their final destinations, lysosomes, plasma membrane, secretory granules, etc.
And as proteins move through these different layers, cis, medial, trans, they get modified.
Yes, sequentially. The Golgi is a major site for processing, especially for modifying the end-linked carbohydrate chains that were initially added in the ER. Different enzymes reside in different Golgi cisternae.
So the sugars get remodeled as the protein passes through.
Precisely. Some mannose residues might be trimmed off. Other sugars, like N-acetylglucosamine, galactose, sialic acid, get added in a specific order by different glycosyltransferases located in the cis, medial, and trans cisternae, respectively. It's like an assembly line for sugar modification.
How does the cargo actually move through the Golgi stack, from cis to trans?
That was a topic of debate for a long time. Two main models were proposed.
What were they?
The vesicular transport model suggested the cisternae themselves were stable, long-lived structures, and cargo moved forward from one cisterna to the next via small transport vesicles butting off one and fusing with the next.
Okay, cargo hops between stable platforms.
The other was the cisternal maturation model. This proposed that the cisternae themselves are dynamic structures that actually mature and progress through the stack. A new cisterna forms at the cis face, then gradually matures into a medial cisterna, and finally into a trans cisterna, eventually breaking down into vesicles at the TGN. The cargo stays within the maturing cisterna.
So the whole platform moves and changes. Which model is favored now?
The evidence now strongly supports the cisternal maturation model. Live cell imaging, for instance, has shown large cargo molecules, like procollagen, which can't fit into small vesicles, progressing through the Golgi stack without leaving the lumen of the cisternae.
So the whole pancake moves and matures.
Right.
But what about those Golgi enzymes? They need to stay in the right place right now.
Exactly. That's where retrograde transport comes in. As a cisterna matures from, say, cis to medial, the resident cis-Golgi enzymes need to be sent back to the new incoming cis cisterna.
How does that happen?
Via small copii-coated vesicles, we'll get to copii soon. These vesicles bud off the maturing cisternae and travel backwards, returning the Golgi enzymes to the earlier compartments where they belong. So, forward maturation of cisternae carrying cargo, coupled with backward vesicular transport of resident enzymes.
A dynamic balance.
Clever. It is. It maintains the biochemical polarity of the Golgi despite the constant flow.
Okay, let's talk vesicle coats. You've mentioned copii and copii. What's the deal with coated vesicles?
Most transport vesicles involved in traffic between the ER, Golgi, and endosomes bud off with a distinctive protein coat on their cytosolic surface.
What does the coat do?
It serves at least two major functions. First, it helps to physically deform the membrane, causing it to curve and pinch off into a vesicle. Second, it plays a crucial role in selecting the specific cargo molecules that should be included in the budding vesicle.
So it shapes the vesicle and chooses the content.
Essentially, yes. There are three main types of coats well characterized: copii, copii, and clathrin.
Let's start with copii. You said it's for ER exit.
Yes, copii-coated vesicles mediate transport from the ER to the ER-GC and the cis-Golgi. They form exclusively at those ER exit sites.
And they select cargo.
How? They capture specific cargo proteins. Some transmembrane proteins have ER export signals in their cytosolic tails that interact directly or indirectly with components of the copii coat. Soluble cargo proteins in the ER lumen often bind to transmembrane cargo receptors, which then get captured by the coat. Even resident Golgi enzymes get packaged this way for their trip to the Golgi.
So it's not just random budding, it's selective loading.
Highly selective, and remember that inherited bleeding disorder you mentioned.
With the ERGC53 protein.
Right. ERGC53 is a cargo receptor in the ERGIC that binds certain coagulation factors. If ERGC53 is mutated, it doesn't get incorporated properly into copii vesicles, the coagulation factors aren't secreted efficiently, and the patient has bleeding problems. Shows the importance of cargo selection.
How's the copii coat assembles and disassembled?
It's regulated by a small GTP binding protein called SAR1. When SAR1 binds GTP, it inserts a little lipid anchor into the ER membrane and recruits the main copii coat proteins, Sec2324 and Sec3331, initiating budding. Later, GTP hydrolysis on SAR1 triggers coat disassembly, exposing the vesicle membrane so it can fuse with its target.
Okay, that's copii for forward transport from the ER. What about copii?
Copii-coated vesicles primarily mediate retrograde transport moving material backwards.
Backwards from where to where?
Mainly from the Golgi cisternae back to earlier Golgi cisternae, as in the cisternal maturation model for returning enzymes, and from the Irgis and Cis-Golgi back to the ER.
Why send things back to the ER?
This is absolutely crucial for maintaining the identity of the ER. Think about it. There's this constant flow of membrane and proteins leaving the ER via copii vesicles. If there wasn't a way to retrieve ER-resident proteins that accidentally escaped, the ER would quickly become depleted of its essential machinery.
Ah, the retrieval pathway, using those return-to-sender signals.
Exactly. ER-resident proteins have specific retrieval signals. For soluble proteins residing in the ER lumen, like the chaperone BP, the signal is typically a 4-amino acid sequence at their C-terminus. KDL, lysas blulu, or a close variant.
KDL means send me back to the ER.
Pretty much. For ER membrane proteins, the signal is often KKXX, 2 lysines near the C-terminus.
How does the cell recognize these signals in the Golgi?
There's a specific transmembrane receptor protein, the KDEL receptor, located mainly in the Ergignum and Cis-Golgi membranes. It has a binding site for the KDEL sequence that's exposed to the lumen.
So it grabs the escaped ER proteins.
Yes, especially in the slightly more acidic environment of the Golgi compared to the ER, which enhances binding. Once bound to a KDEL-containing protein, the KDEL receptor's cytosolic tail interacts with the copii coat proteins,
Loading it onto a copii vesicle going back to the ER.
Precisely. The copii vesicle buds off, travels back to the ER, and fuses. In the neutral pH of the ER, the KDEL protein dissociates from the receptor, which can then cycle back to the Golgi to retrieve more escaped proteins.
The continuous recycling move.
It's essential. If you experimentally delete the KDEL sequence from an ER protein, it gets secreted from the cell. If you add KDEL to a normally secreted protein, it gets returned to the ER. Proof of function.
Okay, we've gotten proteins through the ER and Golgi. Now we're at the trans-Golgi network, TGN, the final sorting hub.
Right. From the TGN, proteins are sorted and dispatched to several destinations. Lysosomes, the plasma membrane, either for insertion or secretion via constitutive or regulated pathways, or endosomes.
Let's focus on the lysosomes. How do those digestive enzymes get targeted there specifically? They travel through the ER and Golgi with all the other proteins, right?
They do initially, but they get a unique tag added in the Cis-Golgi that marks them for delivery to lysosomes.
Not KDEL, something else.
Something else. It involves their N-linked oligosaccharides. A specific enzyme complex in the Cis-Golgi recognizes a structural feature unique to lysosomal hydrolases. This complex then phosphorylates 1 or 2 specific mannose residues on the N-linked sugar chains.
Adding a phosphate group to the mannose sugars.
Yes, creating a unique marker, mannose 6-phosphate, M6P.
So M6P is the send me to the lysosome signal.
Exactly. This M6P signal is then recognized by mannose 6-phosphate receptors, MPRs, which are transmembrane proteins concentrated in the TGN.
MPRs bind to the M6P tagged enzymes. Then what?
The cytosolic tails of the MPRs then interact with adapter proteins, particularly ones called GGAs. These adapters, in turn, recruit the third type of coat protein we mentioned, clathrin.
Ah, clathrin. So M6P leads to clathrin coat formation.
Yes. Clathrin assembles into its characteristic cage-like lattice on the TGN membrane, pinching off clathrin-coated vesicles that contain the MPRs bound to their M6P-tagged lysosomal enzyme cargo.
Where do these clathrin vesicles go?
They travel and fuse with late endosomes.
Not directly to lysosomes.
Not usually directly. They fuse with late endosomes. The environment inside late endosomes is acidic, lower pH. This acidity causes the lysosomal enzymes to dissociate from the MPRs.
The cargo is released. What happens to the receptors?
The now empty MPRs are segregated into vesicles that bud off the late endosome and recycle back to the TGN, ready to pick up more cargo. Meanwhile, the late endosome, containing the released lysosomal enzymes, matures further or fuses with existing lysosomes, delivering the enzymes to their final destination.
Another elegant sorting and recycling system.
Very much so.
And again, what happens if this M6P targeting goes wrong?
That leads to a devastating group of genetic conditions known collectively as lysosomal storage disorders.
Because the enzymes don't reach the lysosome.
Either the enzyme itself is defective or missing due to a mutation, or the targeting mechanism fails. If a specific lysosomal enzyme is deficient, the substance it's supposed to degrade accumulates inside the lysosomes.
Dunking up the works.
Exactly. The lysosomes become engorged with undigested material, eventually disrupting cell function and causing progressive damage to tissues and organs. There are over 40 distinct lysosomal storage diseases known.
You mentioned eye cell disease before as key to figuring this out.
Yes, eye cell disease, inclusion cell disease, was critical. Patients with eye cell disease are deficient in the very first enzyme needed to create the M6P signal, the one that phosphorylates the mannose residues in the cis Golgi.
So no M6P tag gets added.
Right. As a result, their lysosomal enzymes, though synthesized normally, fail to bind MPRs in the TGN and instead get secreted constitutively from the cell via the default pathway.
So the enzymes end up outside the cell.
While the lysosomes remain essentially empty of most hydrolases. Undigested materials accumulate within inclusions inside the cells, hence the name. Studying eye cell disease was how the M6P signal was definitively identified as the lysosomal targeting pathway.
Are there other examples? Tay-Sachs.
Tay-Sachs disease is another tragic example. It's caused by deficiency of an enzyme called hexosaminidase A, leading to accumulation of a specific lipid, GM2 ganglioside, primarily in brain neurons. This causes severe neurodegeneration, developmental regression, blindness, and death, usually by age 4 or 5. It had a higher prevalence in certain populations, but genetic screening has significantly reduced its incidence. Pompe disease involves glycogen accumulation. Gaucher disease involves glucocerebroside accumulation, many different types.
Are there any treatments for these?
Progress is being made, which is hopeful. One approach is enzyme replacement therapy, ERT.
Supplying the missing enzyme.
Yes. For Gaucher disease, for instance, a recombinant form of the missing enzyme, glucocerebrosidase, is produced. It's modified so its carbohydrate chains terminate in mannose residues.
Why mannose, not mannose 6-phosphate?
Because the cells most affected in Gaucher disease are macrophages, and macrophages have receptors on their surface that recognize mannose. So when the modified enzyme, Cerezyme, is infused into the patient,
The macrophages grab it via their mannose receptors.
exactly. They take it up via endocytosis, and it eventually gets delivered to the lysosomes, where it can degrade the accumulated glucocerebroside. It doesn't cure the disease, but it can significantly alleviate symptoms. The movie Extraordinary Measures was based on the development of ERT for Pompe disease.
Wow. What are the other strategies?
Another approach is substrate reduction therapy, SRT. Instead of replacing the enzyme, you use a small molecule drug to partially inhibit the synthesis of the substance that accumulates. Less substrate being made means less accumulates, even with reduced enzyme activity. Miglustat is an example used for Gaucher and Niemann-Pick type C disease.
Different ways to tackle the same underlying problem.
Right. Bone marrow transplantation can also be an option for some, replacing the defective cells with healthy ones. Challenges remain, especially for diseases affecting the brain, as getting therapies across the blood-brain barrier is difficult.
So lysosomes are the recycling centers, packed with acid hydrolases, breaking things down. They also break down old organelles.
Yes, through a process called autophagy, literally self-eating. The cell can engulf old or damaged organelles like mitochondria within a double membrane, forming an autophagosome, which then fuses with a lysosome for degradation and recycling of the components. It's crucial for cellular housekeeping.
And in plants, vacuoles do a similar job.
Plant vacuoles, especially the large central vacuole, have roles analogous to lysosomes containing digestive enzymes, but they also do much more, like storing water, nutrients, and waste products, and maintaining turgor pressure, which provides structural support to the plant cell.
Okay, back to vesicle transport. We know coats help vesicles bud and select cargo. But how does a vesicle know where to go and fuse? It needs to find the right target membrane out of all the membranes in the cell.
Absolutely. This requires incredible specificity. It's a two-step process: tethering and docking fusion.
Tethering first, like throwing out a rope.
Kind of. Initial contact over longer distances is mediated by tethering proteins. These can be long, fibrous proteins like Golgins, which extend out from the Golgi, or large multi-subunit protein complexes. They act like fishing lines or nets to capture incoming vesicles.
How do they know which vesicles to catch?
Specific small GT paces, called Rab proteins, play a key role here. Different Rabs are localized to the surfaces of different membranes, vesicles, and target organelles. They recruit specific tethering proteins, providing an initial layer of specificity. So a vesicle with RabX will preferentially interact with the target membrane, displaying tethers recruited by RabX.
Okay, so tethering brings the vesicle close. Then what?
Then comes the more intimate interaction involving snare proteins. This drives the actual docking and membrane fusion.
Snares. What are they?
Snares, soluble N-ethylmelamide sensitive factor attachment protein receptors, are integral membrane proteins. There are V snares found on the vesicle membrane and T snares found on the target membrane.
Different snares on different membranes.
Yes, and specific V snares interact with specific cognate T snares. When the correct V snare on a vesicle encounters the correct T snare complex on the target membrane, brought together by tethering,
They recognize each other.
they interact very specifically and tightly. Typically, one V snare helix and 3 T snare helices, often 2 proteins contributing the 3 helices, zip together, forming a very stable parallel 4-helix bundle.
What does zipping them together achieve?
This zippering process acts like a winch, pulling the vesicle membrane and the target membrane extremely close together, excluding water molecules from between them. This close apposition destabilizes the lipid bilayers and drives the spontaneous fusion of the two membranes, releasing the vesicle contents into the target compartment or integrating the vesicle membrane proteins into the target membrane.
So snare pairing provides the specificity and the energy for fusion.
That's the current understanding. The specific pairing ensures that, for example, an ER-derived vesicle fuses with the Golgi, not a lysosome. It's a molecular handshake that confirms identity before fusion.
Amazing. Okay, let's switch gears slightly to endocytosis, bringing things into the cell. You mentioned phagocytosis earlier.
Right, cell eating. This is typically carried out by specialized cells, like amoebas hunting for food, or our own immune cells, like macrophages and neutrophils, engulfing bacteria or cellular debris.
How does it work?
The cell extends membrane projections, called pseudopods, driven by actin rearrangement, to surround a large particle, usually 0.5 micrometers. The pseudopods fuse, enclosing the particle in a large endocytic vesicle called a phagosome.
And then the phagosome meets the lysosome.
Yes, the phagosome then matures and typically fuses with a lysosome to form a phagolysosome. The lysosomal enzymes then digest the engulfed material.
But some pathogens can survive this.
Yes, some bacteria have evolved remarkable strategies to thwart phagocytosis. Mycobacterium tuberculosis, which causes TB, gets phagocytosed but prevents the phagosome from fusing with the lysosome, creating a protected niche inside the macrophage. Coxiella burnettii, Q fever, actually thrives in the acidic environment of the phagolysosome. And Listeria monocytogenes can escape from the phagosome into the host cell's cytosol before lysosomal fusion occurs.
Pathogens fighting back at the molecular level.
Absolutely.
What about uptake of smaller things, like specific molecules?
That often occurs via receptor-mediated endocytosis, RME. This is a selective uptake process that relies on specific receptors on the cell surface binding to specific cargo molecules, ligands.
And this involves clathrin again? Those coated pits?
Yes, RME is the classic example of clathrin-mediated endocytosis. The process starts at specialized regions of the plasma membrane called clathrin-coated pits. These are indentations coated on their cytosolic side with a lattice made of clathrin protein.
What does clathrin look like?
The basic building block is a clathrin triskelion, a molecule with 3 legs radiating from a central hub, each leg made of a heavy chain and a light chain. These triskelions have an amazing ability to self-assemble into a polyhedral cage, typically looking like a soccer ball with hexagons and pentagons.
So clathrin forms the cage or basket. How does it connect to the membrane and the cargo receptors?
It doesn't bind directly. It requires adapter proteins, like the AP2 complex for plasma membrane endocytosis, that act as linkers. Adapter proteins bind both to the cytosolic tails of specific cargo receptors and to the clathrin lattice. So receptor-adapter alley-clathrin.
The adapter bridges the gap.
Exactly. This ensures that only receptors, often bound to their cargo, are concentrated into the forming coated pit. As more clathrin assembles, it helps curve the membrane inwards, eventually pinching off a clathrin-coated vesicle containing the receptors and their bound cargo.
And something pinches it off.
Yes, another key protein called dynamin, a GTPase, forms a ring around the neck of the budding vesicle. GTP hydrolysis by dynamin is thought to constrict the neck, leading to membrane fission and release of the vesicle into the cytosol. Shortly after budding, the clathrin coat disassembles, exposing the vesicle membrane for fusion with the next compartment, usually an early endosome.
And specific lipids are involved here too.
Yeah.
Phosphoinositides.
Yes. Phosphoinositides, these specifically phosphorylated forms of phosphatidylinosytyl lipids, act like molecular landmarks on different membranes. For example, the plasma membrane is enriched in PI45P2. This specific lipid helps recruit the AQ2 adapter complex and dynamin to the site of endocytosis. Binding to PI45P2 can also induce conformational changes in AP2, exposing its binding sites for cargo receptors, thus coordinating code assembly with cargo recruitment.
So lipids help define the location and recruit the machinery.
They provide crucial spatial information.
Now, the famous example of RME is LDL uptake, right? The cholesterol story.
Absolutely. The work of Michael Brown and Joseph Goldstein on low-density lipoprotein LDL uptake is a classic Nobel Prize-winning story that beautifully illustrates RME and its medical relevance.
What was the disease they studied?
Familial hypercholesterolemia, FH. Patients with FH have extremely high levels of blood cholesterol carried in LDL particles, leading to premature atherosclerosis and heart attacks, sometimes even in childhood.
What was their key discovery?
They were studying fibroblasts, skin cells, from normal individuals and FH patients. They found that normal cells could take up LDL from the medium, and this uptake suppressed the cell's own cholesterol synthesis. FH cells, however, couldn't take up LDL effectively and continued to synthesize cholesterol even when LDL was abundant outside.
Suggesting a defect in LDL uptake.
Exactly. They then used radioactively labeled LDL and showed that normal cells had high affinity binding sites for LDL on their surface, whereas cells from severe FH patients had virtually none. This pointed strongly to a defect in the LDL receptor.
So FH patients lacked or had defective LDL receptors.
That was the core finding. They confirmed this visually using electron microscopy with LDL particles linked to electron-dense ferritin. On normal cells, LDL ferritin bound specifically to those clathrin-coated pits. On FH cells lacking the receptor, no binding occurred at coated pits, even though the pits themselves were present.
And then there was the patient JD.
Yes, the JD mutation was fascinating. This patient's cells had LDL receptors that could bind LDL normally on the cell surface, but they couldn't internalize it. The receptors weren't localized in the coated pits.
What'd that show?
It showed that the receptor protein itself needs a signal, specifically in its cytoplasmic tail, that allows it to interact with the adapter proteins, like AP2, and get incorporated into the forming clathrin-coated pit. The JD mutation was in this cytoplasmic tail signal. It separated binding from internalization.
So RME requires both a receptor to bind the cargo and a signal on the receptor to link it to the clathrin adapter machinery.
Precisely.
And now we can even watch single LDL particles being internalized.
Yes, with modern fluorescence microscopy, you can label clathrin with GFP, green, and LDL particles with a red fluorescent dye. You can literally watch a single red LDL particle being captured by a transient green clathrin-coated pit, internalized into a vesicle that quickly loses its green coat and then move deeper into the cell. It's quite beautiful to see.
So once the LDL particle is inside, in an endosome, what happens?
This depends on the type of receptor. LDL receptors are considered housekeeping receptors. Their main job is to bring essential cargo, cholesterol and LDL, into the cell.
Okay.
In the mildly acidic environment of the early endosome, the LDL particle dissociates from the LDL receptor. The receptor is then sorted into vesicles that bud off the endosome and recycle back to the plasma membrane, ready to bind more LDL.
So the receptor is reused. What about the LDL itself?
The LDL particle stays in the endosome, which matures into a late endosome and eventually fuses with a lysosome. There, the LDL particle is broken down, releasing the cholesterol for the cell to use.
Efficient delivery and receptor recycling. What about other types of receptors? Signaling ones.
Signaling receptors like the epidermal growth factor, EGF receptor, behave differently after endocytosis. When EGF binds, the receptor is internalized via RME, but often instead of recycling, the receptor itself is targeted for degradation in the lysosome.
Why destroy the receptor?
This is a mechanism called receptor downregulation. By destroying the receptors after they've signaled, the cell becomes less sensitive to further stimulation by that ligand. It's a way to terminate or attenuate the signal. Often, these receptors get tagged with ubiquitin after activation, which signals for them to be sorted into internal vesicles within the late endosome, forming a multivesicular body, ensuring they get degraded along with the ligand in the lysosome.
Two different fates after endocytosis: recycling or degradation.
Depending on the receptor and its role.
And the LDL particle itself, what's it made of?
It's basically a transport package for cholesterol. It has a core containing hundreds of cholesterol molecules esterified to fatty acids, making them very hydrophobic. This core is surrounded by a shell of phospholipids and unesterified cholesterol, plus one very large protein molecule called apolipoprotein B100, ApoB.
And ApoB is what the LDL receptor recognizes.
Yes, ApoB is the ligand that binds specifically to the LDL receptor on the cell surface.
Understanding LDL has obviously been huge for understanding heart disease, right? Atherosclerosis.
Immense. The model is that high levels of LDL in the blood can lead to LDL infiltrating the artery wall, where it can become oxidized. Macrophages in the artery wall engulf this modified LDL, but they can't process it properly and become engorged, turning into foam cells.
Foam cells. Sounds bad.
They are. They contribute to the formation of atherosclerotic plaques, fatty deposits that narrow the arteries. These plaques can become unstable, rupture, and trigger blood clots, leading to heart attacks or strokes.
And cholesterol-lowering drugs often target this pathway.
Many do. Statins, for example, inhibit the cell's own cholesterol synthesis, which leads to upregulation of LDL receptors, helping to clear more LDL from the blood. And newer drugs, the PCSK9 inhibitors, are really interesting.
What do they do?
PCSK9 is a protein that normally promotes the degradation of LDL receptors. By inhibiting PCSK9, these drugs prevent LDL receptor destruction, leading to more receptors on the liver cell surface, lower blood LDL levels, and reduced cardiovascular risk, directly manipulating the receptor life cycle.
Amazing how understanding the basic cell biology of receptor trafficking leads to powerful therapies.
It's a perfect example of that bench-to-bedside connection.
Okay, one last major topic: post-translational uptake. We mentioned that some organelles import proteins after they're made in the cytosol.
Right. While the ER handles co-translational import for the secretory pathway, organelles like the nucleus, mitochondria, chloroplasts, and peroxisomes rely primarily on importing fully synthesized proteins from the cytosol.
Do they use similar principles, like sorting signals?
Yes. The general themes are similar. Proteins destined for these organelles are synthesized on free ribosomes and contain specific sorting signals or targeting sequences within their amino acid structure. These signals are recognized by receptors located on the surface, usually the outer membrane, of the target organelle.
And then they get transported across the membrane.
Yes, through specialized protein translocation channels, or pores, specific to each organelle. Often, the proteins need to be unfolded or kept in an unfolded state by cytosolic chaperones to pass through these channels, and then refolded by chaperones inside the organelle.
Can I look at chloroplasts as an example?
Sure. Most chloroplast proteins are actually encoded by nuclear genes, synthesized in the cytosol, and then imported.
How do they get targeted directly?
They typically have an N-terminal targeting sequence called a transit peptide. This transit peptide is recognized by receptor proteins on the chloroplast outer membrane, part of the TOC complex, translocon at the outer chloroplast membrane.
So TOC is the entry gate.
Right. The protein is then passed to the TIC complex, translocon at the inner chloroplast membrane, and translocated across both membranes into the chloroplast's internal space, the stroma. This requires energy, often ATP hydrolysis.
And chaperones are involved.
Yes. Chaperones like HSP70 and HSP90 in the cytosol help keep the protein unfolded, and chaperones like HSP60 inside the stroma help it refold correctly after import. Once inside the stroma, the transit peptide is usually cleaved off.
What if the protein needs to go deeper into the thylakoids?
Proteins destined for the thylakoid membrane or the thylakoid lumen have an additional signal sequence, a thylakoid transfer domain, downstream of the stroma-targeting transit peptide. After the transit peptide is cleaved in the stroma, this second signal directs the protein across the thylakoid membrane via yet another set of translocation pathways.
Multiple signals for multi-step targeting.
It's a very precise hierarchical targeting system ensuring proteins reach their correct subcompartment within the chloroplast. Similar principles apply to mitochondrial and peroxisomal import, though the specific signals, receptors, and translocons differ.
Wow. Okay, that brings us almost to the end of our deep dive. We've covered an incredible amount of ground.
We really have. from the initial synthesis in the ER, through the processing in the Golgi, the sorting at the TGN, the delivery via vesicles, the retrieval pathways, endocytosis, lysosomal function, quality control. It's just staggering.
The sheer complexity, but also the precision and interconnectedness of it all. Every step seems so tightly regulated. Signals, receptors, coats, rabs, snares.
It has to be, doesn't it? The fidelity is amazing. And reflecting on how scientists figured this out using those ingenious techniques like pulse chase, GFP imaging, fractionation, genetics with mutants, RNAi, even studying human diseases, it's a real testament to scientific creativity and persistence.
Absolutely. And for you listening, this isn't just abstract biology. Understanding these pathways, as we've seen, is fundamental to understanding health and disease. From cystic fibrosis to lysosomal storage disorders to heart disease, it all comes back to these basic cellular processes.
Knowing how the cell's internal traffic system works allows us to understand what happens when it breaks down and, crucially, how we might be able to fix it, like with enzyme replacement therapy or PCSK9 inhibitors.
It leaves you with some big questions, though, doesn't it? I mean, considering the sheer volume of traffic inside a cell, all those proteins, lipids, vesicles constantly moving, how does it maintain such near-perfect fidelity? How does it really prevent those catastrophic traffic jams or missed deliveries that could lead to disease?
That's a great question. The level of regulation and coordination is almost beyond comprehension. And thinking forward, how can we learn even more about this cellular GPS system? Could we perhaps hijack it ourselves in even more sophisticated ways for future therapies, delivering drugs precisely where they need to go by tapping into these natural sorting mechanisms?
Lots to think about. Well, that concludes our detailed look into cytoplasmic membrane systems. We've tried to cover the key structures, functions, trafficking pathways, and experimental discoveries as laid out in Karp's.
It's a dense chapter, but hopefully breaking it down this way helps make sense of the cell's incredible internal logistics.
Thank you so much for joining us on this deep dive.